Thank you.
Good day, ladies and gentlemen, class, Dr. Campbell, and audience of YouTube. Today, our presentation will be on nanotechnology in immunology.
What is nanotechnology? According to the U.S. National Nanotechnology Initiative, nanotechnology is understanding and control of matter at dimensions of roughly 1 to 100 nanometers, where unique phenomena enables novel applications. Now we will look at a brief history on nanotechnology. In 1959, Dr. Richard Feynman provided the first inspiration for nanotechnology. His lecture was based on "There is plenty of room at the bottom," which he described as there is plenty of room by decreasing the size of things in a practical way. He said that the ability to manipulate individual atoms and molecules by using one set of precise tools to build and operate another proportionally smaller set. In 1974, the term nanotechnology first derived by a Japanese scientist, Dr. N. Taniguchi, who described nanotechnology as the process of separation, consolidation, and deformation of material by one atom or one molecule. Later, in 1981, the invention of the scanning tunneling microscope by nanocrystalline and semiconducting quantum dots in a glass tube. Discovery of fullerenes was named after Buckinghamshire Fuller. It is a hollow spherical ellipsoid or tube of carbon, which is similar to graphite, but it is in a pentagonal ring, which prevents it from becoming planar, and it gives it different shapes. One of the fullerenes are spherical and another is cylindrical. Spherical fullerenes, or also known as buckyball, is made up of carbon-60. It resembles a soccer ball in the shape and it composed entirely of carbon. While spherical fullerenes, also known as bucky tubes, and recently bucky egg was also discovered. In 1986, atomic force microscope was first discovered, which is able to view, measure, and manipulate materials to nanometer size. And in 1989, the first xenon atom was used to spell the M logo by manipulating atoms of nanotechnology. In the 1990s, there was a boom in nanotechnology. There was government support and nanotechnology companies also developed. Through 1990 to 1993, the nanostructural catalyst material was used for crude oil refining. In 1999 to early 2000, nanotechnology began appearing in the marketplace, such as in flying straight golf balls and dent and scratch resistant automobile bumpers.
Benefits of nanotechnology in immunology. Successful applications of nanotechnology in the field of immunology will enable new generations of vaccines, adjuvants, immunomodulatory drugs for a range of infectious diseases and non-infectious diseases, as well as new immunomodulatory agents such as nanostructures. Here we see some examples of nano devices that can be commonly used in immunology. There are 4 general mechanistic pathways in which immunology interacts with nanotechnology. Delivery of antigens. Nanoparticles can be used to carry antimicrobial agents. The carrier hides and protects the molecules from degradation in the body and allows it to be delivered to specific target cells in a controlled manner. Special carriers can be designed to carry therapeutic drugs across the membranes into specific intracellular compartments such as the blood-brain barrier. This can be used for the treatment of many intracellular infections, including fungal and parasitic infections, salmonella and tuberculosis. Nanoparticle carriers can be metallic or lipid-based. Depot effect. Nanoemulsions are oil and water droplets. They are high in energy, thermodynamically driven to fuse with lipid-containing organisms. The electrostatic attraction between the cationic charge of the emulsion and the anionic charge. When fusion occurs, the active ingredients inside the particle and the energy released in the fusion process destabilizes the lipid membrane, resulting in cell lysis and death. Repetitive antigen display. The spatial organization of the antigen on the particle surface facilitates B receptor co-aggression, triggering an activation, thereby producing high affinity antibodies. Cross-presentation. Exogenously acquired antigens are processed into MHC class I pathways. Nanoparticle-mediated leakage of antigens into the cytosol after they have been taken up by the phagosome. Applications and uses of nanotechnology in immunology.
Right. There are diagnostic techniques that are used. We have a high degree of sensitivity that promote detection of biomarkers. This high degree of sensitivity is different to wavelength of light, which is adjusted by the number of atoms and the size of the quantum dots. The non-invasive and painless diagnostic techniques are normally conducted by nanoparticles being electrostatically and biochemically aligned according to diagnostic procedures to be conducted. There's genetic testing for individual therapy sessions. This is like therapy based on the genome or genetic makeup of the individual. Targeted drugs. The nanoparticles containing drugs are coated with targeted agents, example conjugated antibodies. The nanoparticles circulate through the blood vessels and target cells. Drugs are released directly into the targeted cells. Now, nanoparticles are submicron sized polymeric colloidal particles with therapeutic agents of interest. They can be either encapsulated or conjugated on the surface. They are highly versatile in that they overcome physiological barriers and guide the drugs to specific cells. They control release patterns of drugs and drug levels for a long time.
Regenerative medicine. Current studies are working on biomimicking cell membranes, polymers with programmable conformations, targeted cell implantation, and control of implant rejection. Some researchers are currently working with nanotechnology to treat cancers. They have found that it has been efficient in targeting cancer cells, reduced side effects to the patient, and had a greater drug retention. Current research is emphasizing on siRNA and penetrating peptides, doxorubicin delivery, and nanomaterial drug combination. Another example is with the treatment of mucosal immune disease. They have found that it has been an efficient drug targeting system, reducing side effects and optimizing the concentration of drugs used. Current research is emphasizing on biodegradation and biocompatibility of materials that are capable of transporting anti-inflammatories, nucleic acids, and other biomaterials.